Hello everyone, welcome to the IMAG Multiscale Modeling webinar series, and I'm pleased to introduce Dr. Ed Sander, a new multiscale modeling participant who will be presenting his work to us here today. Dr. Sander will be talking about controlling the mechanobiology of cutaneous wounds to reduce hypertrophic scarring. So take it away, Ed. Thanks.
Okay. Thanks very much to everyone for spending some time with me today. So the title of my talk is the title of the RO3 I was recently awarded, called Controlling the Mechanobiology of Cutaneous Wounds to Reduce Hypertrophic Scarring. And what I want to do in this webinar is just briefly lay out what multiscale mechanical interactions are, so we have a baseline for this conversation. And then what I want to do is talk about some previous work, two projects that I was involved in as a postdoc at the University of Minnesota with Victor Barocas, who's a consortium member, and Bob Tranquillo, that I think will help explain sort of the frame of mind for viewing this proposal and kind of lead you to how this proposal came into being. Then what I'll do is I'll just briefly introduce some ideas of wound healing, talk about the grant proposal, the aims and the current work and the future work involved in that. And so multiscale mechanical interactions are physical interactions that span across scales within soft tissues, starting from the extracellular matrix and the individual collagen fibers, which you can see on the right-hand side here. These collagen fibers are organized into hierarchical structures, such as collagen fiber networks, that are in increasing levels of organization as you move up through the different scales of the whole tissue. So what you can see here is an example of a cell-compacted collagen gel, or a tissue equivalent. And so, you know, we know that the composition and the organization of the microstructure of this gel is deterministic of the mechanical properties and its function. And so we have mechanical multiscale interactions coming from that direction. But the other component of this is that the loads and constraints that are applied at the tissue level are transmitted down into the extracellular matrix, where they can provide signaling mechanisms that alter or affect cellular behavior, which in turn can result in a change in the composition and organization of the microstructure, and hence the mechanical properties. And so in this simple schematic, you can see these ideas laid out, showing, again, the tissue which exists on the millimeter or centimeter scale, the loads and constraints that may be applied to it that are transmitted into the extracellular matrix, which can exist at the micron or nanometer scale, how those forces and mechanical cues are experienced by the cell, which can result in matrix synthesis or degradation, which again changes the matrix composition and organization, and hence the tissue properties. And so these multiscale mechanical interactions, for me, have been of primary interest in the context of engineering tissues and how we can make the remodeling process that we're trying to direct better to produce things that are more like native tissues. And with that in mind, there's sort of two broadly classified areas that I think are required that we understand, and that are the structure-function relationships, such as how is the extracellular matrix put together, and why is it put together that way. And if we look at a native tissue, we can think of those relationships as the blueprints or the design targets for our engineered tissues. And then the second component of that is the growth and remodeling processes. So how are the cells coordinating construction of the tissue? How are they maintaining the extracellular matrix? And how are the cells responding to changes in the extracellular matrix? And both of these things are dependent on these multiscale mechanical interactions, this communication of mechanical factors across scales, and they're involved in a variety of biological phenomena, including development, disease, and damage, as well as in applications like engineering tissues. And so to understand these things better, while I was a postdoc, I worked on two projects. And the first project that I want to describe in more detail is developing a computational framework for really understanding how these components come together to give an engineered tissue its functional properties. And so this is developing an image-based multiscale model that is composed of three primary components. The first is our test sample, which in this case is a cell-compacted collagen gel, and I'll describe that in more detail in the next slide. The second primary component of that is, of course, the computational model. And the third component of that is the experimental system that we use that allows us to simultaneously mechanically test and image the gel so we can get measurements of how the microstructure reorganizes dynamically in response to loading. So our test samples, these cell-compacted collagen gels, are a nice system for us to use because they're relatively simple compared to soft tissues, and they contain some of the essential features that are involved in these multiscale mechanical interactions. And so what you can see here is a cross-shaped Teflon mold that we refer to as a cruciform. And what we do is we put a solution of homogeneously dispersed fibroblasts and collagen into the space here. It fills the space and it infiltrates into these Velcro strips. And then what happens over several days of culture is that the cells start to attach the fibers of the gel, compact it, bring it in, and reorganize the matrix. And so you can see that the gel has started to pull away from the sides of the Teflon mold, except in the places where it's constrained. And if you look at a fiber alignment image, which I'll describe in a little more detail in the next slide, you can get a measurement of the orientation and strength of alignment of the microstructure. And so here you can see for this asymmetric cruciform where we have a horizontal axis that's twice the width of the vertical axis, we've got these strong regions of alignment in the arms, and then in the center region here, we have moderate horizontal alignment and some regions of isotropy in these particular locations. And what happens is when these structures are created to generate these fiber alignment patterns in the tissue, it arises from the interplay of multiscale mechanical interactions between cell compaction of the gel, the geometry of the gel, and the constraints on the gel. So we can manipulate these things to create our test samples. So the polarized fiber alignment imaging, what it's doing is transmitting light through the sample and allowing us to measure the direction-dependent birefringent properties of the sample at each pixel. And what this represents is a measurement of a collection of fibers at that location. Now, if you look at the images here on the left-hand side, this is the initial reference configuration. And the regions that are bright correspond to strongly aligned regions, and the dark region in the center corresponds to an isotropic or less aligned region. There are also a couple of artifacts here which are actually markers for tracking strain, so just ignore those. The important thing to recognize is in the deformed image where we've stretched the vertical axis and kept the horizontal axis stationary, there is significant restructuring, reorganization that's occurring in response to the load. And we can get measurements of the strength of that realignment by correlating that to this retardation value. And we can also get the primary direction of orientation, which you can see in the maps below. So we have the ability to acquire this information. We have a computational modeling framework that's been developed over several years. What it is that we use volume averaging in concert with finite element simulations. So what you can see here is a finite element domain that represents one quadrant of a cruciform, assuming symmetry. And what's different here is that instead of using a constituent equation at the continuum level to relate displacements to the stress, instead we solve a set of microscopic fiber network problems at the location where the stress is needed to get the finite element solution. So we have here are a collection of collagen fiber networks that are contained within a box that we sometimes refer to as a representative volume element. Each of these boxes is centered at an integration point in an element. And what happens is when we deform the finite element domain, the nodes of the element change position, which causes the boundaries of the RVE to change. The fibers that are connected to those boundaries then become stretched. They pick up forces in accord with the constituent equation that we specify at the fiber level. Those forces are transmitted to other fibers in the network via crosslinks that we handle as ball joints. And the whole system of fibers reorganizes in order to satisfy force equilibrium. We then volume average over the local stresses to get a macroscopic stress tensor, and that's what we then use in the macroscopic stress balance in obtaining the solution. So we have all of these things happening across scales, and we have iterations going back and forth until we get convergence throughout the model. So as a result of representing the microstructure with this set of microscopic fiber networks, we're able to relate the tissue's behavior directly to fibromechanical properties and network structure. And to give you a better sense of what that looks like before I get any further, here you can see one of these fiber networks reorganizing in response to this in-plane stretch. And you'll see that the box is shrinking in the Z direction. That's not from compression, that's just the Poisson effect from the in-plane tension that we're applying at the macroscopic level. Now, in this particular study, one of the things we were very interested in was capturing the heterogeneity of fiber alignment that exists in these samples, and then creating these models that match the initial conditions, simulating the experiment, and see how well the models predict what we observe experimentally. And so to do that, we created masks by projecting the area of the finite element onto the imaging data so that we could extract out an average orientation and strength of a fiber alignment associated with the microstructure at that particular location in the experiment. And I'm not going to get into the details of how we generate these networks. The important points are that we make the projections of our networks match the imaging data because the imaging data represents values that are projected through the tissue. And we use this thing called an orientation tensor whose eigenvalues and eigenvectors correspond to the magnitudes and directions of fiber orientation of our networks. We can then go through a process where we set limits on an isotropic versus a completely aligned network, and then create networks that match our experiment within a given tolerance. And so in this image here, you can see the model networks in red overlaid on the measurements in the experiment in white. And so this is showing a simulation for an off-axis hold test. This is where the horizontal axis is stationary and we deform the vertical axis. And what you can see is that the model does a good job, which is shown in red, of predicting the rotation and stretch that we see experimentally. You can see the force extension data next to that, and what we do is fit that by altering the parameters A and B at the fiber level for the constitutive equation. We then take these gels and deform them in different ways. This is an equibiaxial stretch test, and we find that we are able to do a reasonably good job of predicting the communication of mechanical interactions across the scales and in predicting the force response. Now, the real utility, I think, of these kinds of models at this point are that they help us really develop conceptual ideas about the importance of heterogeneity in the microstructure and how these multiscale mechanical interactions arise and how they might dictate what the local mechanical environment is for a cell. And so to help you hopefully see that better, I'm showing you now what certain networks in the microstructure look like. So this is a aligned fiber network in the vertical axis of the arm. It starts off aligned, and then for an off-axis hold test where the horizontal axis again is stationary and the vertical axis is being stretched, you can see that the fibers rotate down and stretch. This is a histogram down here of the individual fiber stretch ratios of the network, and you can see that most of them are below what you calculate as a macroscopic stretch ratio if you were basing it off the macroscopic displacements. If you look at the same network for equibiaxial stretch, where now we're also deforming the horizontal axis, the kinematics of the fiber reorganization is similar, but it picks up higher loads, and the fibers in the network are stretched more, and so you end up having a collection of those fibers that experience stretch levels that are more like the macroscopic level or higher. Now, that happens because of other networks throughout the model also reorganizing in response to the displacements. And here you can see a network from that center region. And this network starts off with some moderate alignment in the horizontal direction. And so for this off-axis hold test, where we again stretch this axis and keep this one fixed, the fibers are free to rotate before they then stretch and pick up forces. And you can see that in the histograms where the fiber stretch ratios are fairly low. And again, for an equibiaxial stretch case, you can see that the reorganization of the fibers of the network is very different, as you might expect, and that there are much higher fiber forces that are being generated here because the fibers don't have the same kind of rotational freedom because we're doing equibiaxial stretch now. And just to help you see this a little better, these are movies of the individual fiber networks I just spoke about. The way to look at this is the initial configuration of the mesh, and this is increasing off-axis stretch, which would correspond to this direction on the networks. this is the equibiaxial stretch where both axes are being stretched. So this is horizontal and this is vertical direction. And what you can see is that the fiber kinematics is heterogeneous and quite complex. And the thing that I want to emphasize here is that, we expect there to be differences here, and as a result of, the initial microstructures configuration, the mechanical loads that we're applying. But the way the structure reorganizes and deforms as we stretch things is very much dependent on how all the networks throughout the model are simultaneously reorganizing as well. So we have this ensemble response that develops through the interaction of all of these different microstructures that are heterogeneous in the model, and that's important to consider because our soft tissues are heterogeneous as well. Okay, so the second component that I want to talk about previous work is experimental in nature, and what that involves is trying to understand the role of these multiscale mechanical interactions in the remodeling process when you're trying to engineer a tissue. And again, I'm spending some time on these issues because they tie into the aims and the scope of the proposal. So this is work that I was doing primarily in Bob Tranquillo's lab, and what you can see here is that Dr. Tranquillo's group has been engineering arteries and heart valves using fibrin as a scaffolding material for quite some time. And the general idea there is that if you control the way the gels compact in the initial phase, you can then create organization and alignment of the cell-produced matrix in a way that confers better mechanical properties to the tissue. And so a simple example of that is if you take this tubular cavity that has this non-adhesive mandrel or rod in the center, and you inject a solution of monomeric fibrinogen and cells like fibroblasts, then what happens over a few hours to a day or two is that the cells start to compact down the gel, which slides along the mandrel. In the process, it generates this circumferential alignment. And so what then happens is this fibrin degrades, and the cell-produced matrix follows this initial alignment pattern. So the fibrin here is acting as a template that's guiding the organization of the collagen and other extracellular matrix that's produced. And I think it's important to point out that if you're not familiar, fibrin is the primary load-bearing protein that you have in a clot when you have hemostasis. So the fibrinogen polymerizes into fibrin and basically starts hemostasis for you. So you can use this sort of design strategy for engineering tissues to produce things that are more sophisticated or complicated in their alignment pattern. This is an example of a heart valve leaflet. And we get these compositional changes, but we also direct these organizational changes. And as a result of that, we get much better mechanical properties for these engineered constructs than we do if we didn't take into consideration the organizational aspects of this. But there's a lot about this process that we don't really understand, particularly how these multiscale mechanical interactions develop and sort of give rise to remodeling events in different locations in the tissue. And so what I was interested in working on here was asking the question, how does the initial fiber alignment that develops from these interactions affect the remodeling process? And so what we're doing here is, again, going back to our cruciform test system, but what's different here is these are not collagen gels, they're fibrin gels. And if you look at the fiber alignment images for these gels, you can see these differences in the alignment patterns. So on the left, we have a symmetric cruciform where the horizontal and vertical axes are the same width, 8 millimeters. And what you can see, just like before, is that the arms are strongly aligned, but there's this large center region that is less aligned and close to isotropic. In contrast, with the asymmetric cruciform that I just finished describing for the collagen gel system, you again have the strong alignment in the arms, but now the center region has this moderate alignment in the horizontal direction, and this region of isotropy has split and shifted along the vertical axis. And just to say it one more time, these alignment patterns are dependent on these multiscale mechanical interactions that arise from the interplay between cell compaction of the gel, the geometry of the gel, and the mechanical constraints on that gel. And so there are a lot of things we looked at in this study, but the most important thing that's relevant to the proposal is we culture these things over a 10-week period and look at how the remodeling process is unfolding and how the mechanical properties are changing. And what we found is that we get the most collagen production in these narrower arms of the cruciforms, this orange region, than in any other region of either of the cruciforms. And so that was an interesting result for us, particularly because there are actually more cells, total cells in the wider arms, because these things are made at the same cell concentration. And if we normalize by total protein, we get the same thing. And if we just look at the total amount of collagen, we often get more just total collagen in these arms, or at the very least an equal amount. And so to try to better understand what's going on there, we did some simulations using something called the anisotropic biphasic theory. And I won't get into the details of that, but the important part of that is that with the model, if you specify a cell concentration, a geometry, and the constraints, what the model will do is predict what the final alignment pattern will be for that construct. And here you can see what the stress maps look like. And what we find is that the predictions of fiber alignment match very well with what we see experimentally. And if we look at the stresses, we see a correlation between the regions of high stress, if you ignore the edge effects, and the locations where we see abundant collagen production. So in particular here you can see this yellow region, and that's where we get the most collagen per cell. So this collagen production appears to be correlated to the local mechanical environment that develops from these multiscale mechanical reactions. And this is just in a static system. We're not stretching it or anything like that. So it seems like these cells are communicating across the tissue and setting up a loading environment that is heterogeneously affecting cell behavior in terms of the remodeling process. Okay, so all of this now relates to this idea of wound healing. And so just so we are all on the same page, this is just a schematic showing a nice drawing of cutaneous wound healing. So you have, injury to the skin, you have hemostasis and the formation of fibrin clot, and you have infiltration of a bunch of other cell types in addition to degranulation and contraction of platelets. And you can watch this process unfold until you reach a final healing period and sort of classify different stages. A common way to do this is to divide it into three stages as an inflammatory phase, a proliferative phase, and a remodeling phase. And this schematic gives you just a general sense of, you've got leukocytes that are coming in initially, then you've got the fibroblasts and so on. And this axis is days post-wounding. You then have different components of the matrix that are getting produced. So we have this initial production of fibronectin and type 3 collagen that's gradually replaced by type 1 collagen. And here you can see conveniently an increase in the wound breaking strength, which is strongly correlated with the amount of type 1 collagen. So that's wound healing in general. Now, if you have some kind of surgical defect, you can close the wound or leave it open, either primary or secondary intention healing. In this case, you've got sutures to close the wound. When the sutures are removed, there is a wound healing response that's ongoing. This will likely resolve as a small scar that's flat, what would be considered normal scarring. However, it's possible that what can happen, and this is not related to, this is a separate image, and it's not related to the patient series I'm showing you here, but what can happen is you can have excessive collagen production and develop these raised scars that stay within the wound boundaries, and that's referred to as hypertrophic scarring as opposed to normotrophic scarring. And so this is a major clinical problem that can, it's estimated to affect around 4 billion annually to treat in the US alone. And as I said, it's characterized by excessive fibrosis and collagen production, and it can result in disfigurement, distress, pain, and loss of function, either through joint contracture or loss of mobility. So it's a big problem, and there are a number of different clinical treatments that are in practice right now, and just a few of them are things like corticosteroid injection, silicone gel sheeting, use of paper tape, pressure therapy. And what's interesting is that when they are able to resolve fibrosis with these techniques, and it doesn't always work, a lot of these techniques are believed to have a mechanical basis underlying how they're reducing fibrosis, which is consistent with what's long been known in dermatology and making surgical incisions, and that is that the tension surrounding the wound site can play a prominent role in fibrosis. And that's why surgeons will try to make incisions following these things called Langer's lines, because you have less tension on the wound if it's oriented in that direction. Now, some recent work by Gertner and several colleagues at Stanford that I think is very interesting and pretty exciting is they took this idea about reducing tension in the wound site and tried to rectify that by using a load-bearing polymeric device and applying it over the wound site to reduce or change the mechanical environment. And what you can see down here is a grid that's on, I think it's on a pig, and then you can see when they put this device on it, what it does is it basically brings the tissue together and puts it in compression. And so you can imagine that you'd make some kind of surgical incision around the horizontal and then put this on top, and it would put the wound in compression as opposed to tension. And in addition to animal studies, they tried this with abdominoplasty on patients where they treated one side with this stress shielding device and left the other side the way they would normally deal with the wound healing process. And this is just showing a couple of examples of within patient response, and you can see what I think is a pretty dramatic reduction in scarring that's occurring by just using this polymeric device. Now, they did report that the healing response was variable. That's to be expected because there are a lot of different factors at play, including genetics, the patient's health, the location of the wound, and many aspects like that. And so what I'm interested in is really understanding these mechanisms and how they relate to the development of fibrosis. And so to do that, we need to get a more detailed view of the underlying mechanisms and maybe how that gives rise to the variability. And so based on my previous experience, what I wanted to focus on was this notion of these multiscale mechanical interactions that develop between the cells, the matrix, and the wound boundaries. And so you can think of this as a feedback loop. And so here's just a simple schematic. And so it will start with hemostasis and the formation of a fibrin clot. You've got some mechanical loads, the wound geometry that affect the mechanical environment, the stress and strain. You also have internal tension that's developing from cells themselves. That's affecting and signaling the fibroblasts. You have growth factors that are also involved. Those can lead to synthesis or degradation of extracellular matrix, which then changes the composition and the organization and alignment of the matrix that's in there, which then feeds back, changing the mechanical environment, perhaps signaling more or less of a certain type of growth factor, like, for example, TGF-beta. And this process will continue to cycle around until you get to some sort of outcome, which in the ideal limit would be total regeneration. The other side of things would be exuberant scarring. And what we want to do is gain control of this loop, identify what factors are part of this loop so that we can modify and direct the range of scarring outcomes to be more in line with regeneration. And so the central hypothesis of this proposal is that these multiscale mechanical interactions amongst the initial organization of the clot, the constituent cells, and the external force constraints are deterministic of the fibrogenesis that controls scar phenotype in a healed surgical wound. And the sort of difference in thinking here as opposed to what I showed you in the previous slide is that in addition to the wounding that's occurring, the mechanical environment occurring around the wound site, the idea here is that the initial conditions that come together in hemostasis and forming the clot basically frame the remodeling outcome space that you can have. And so as a corollary of this hypothesis, if we can control these interactions, then fibrosis can be minimized and tissue repair can be closer to the unwounded skin. And so the short-term goal of this proposal then is to develop a new model system for studying the mechanobiological basis of scar formation that combines experiments and computational models in a manner that is very similar to what I showed you earlier with the sort of instantaneous multiscale mechanical interaction relationships within a collagen gel. And the long-term goal is to eventually develop predictive models of wound healing so that we can devise patient-specific strategies to minimize scar formation, where we can take all of these other factors into account, perhaps even recommend different drugs to accompany anything we do to modulate the mechanical environment of the wound site itself. And so the first hypothesis for the proposal then is to test the hypothesis that multiscale mechanical interactions control fibrogenesis during the remodeling process in an in vitro setting. And what this really means is that we need to characterize and quantify these fibroblast extracellular matrix mechanical interactions, how they control the remodeling process, and how multiscale mechanical interactions come to play in directing that remodeling process in terms of the initial fiber alignment patterns, the cell spatial distribution, the geometry, and the load. And then once we've got that solidified, what we want to do is start perturbing the environment and seeing how the external loading environment can alter this process at more of a mechanistic level. That first aim can be thought of as the experimental side of things. The second aim then is to develop an image-based multiscale mechanical model of the wound site that's based on the in vitro data we're collecting from these fibrin gels. And so to give you a sense of what we're doing experimentally, this is showing in the lab what we're using right now. We have an inverted epifluorescent microscope with DIC capabilities, and we have an environmentally controlled bioreactor that is equipped with a load cell and a linear actuator so that we can measure force and deform our gels when we want to. And when we seal it up, we also have inlet and outlet ports that we can use to replenish the medium, take medium samples if we want to. And of course, this is all mounted on a microscope so that we can actually do extended time-lapse imaging of cells in this system. And so this is just showing mouse L929 fibroblasts, which we were using as a test cell to just kind of get started with developing the system. And what you can see here is that the fibroblasts are extending processes into the surrounding fibers of a collagen gel. Hopefully you can see that there's a little bit of displacement occurring here. It turns out the mouse L929 fibroblasts are not nearly as active in terms of contracting the matrix as human fibroblasts or fibroblasts from other animals. And anyway, before I get to the next slide, one of the issues here with using homogeneously distributed cells like this is that it's difficult to pinpoint, because there's so much going on, which cells are interacting with which. So we've decided to step back and move to a simpler system, and the inspiration for this was derived from some work by Sonny and Howard about 10 years ago that is also built off of some even earlier work. And what they did in this system was they took a collection of cells and put them down in these balls that they call explants, and that allows you to watch how the explants collectively contract and reorganize the matrix around them. And there's several advantages to doing this. One is that it allows you to more precisely control where these cell populations are, and therefore better see how they're reorganizing things. It also allows you to keep things relatively planar, and it has some other advantages too that are related to modeling. And so we've adopted this to get started, where we make these explants and we watch how they reorganize the fibers between them. Here you can see in a fibrin gel the fiber alignment that's developing, forming this sort of triangle pattern. This is sometimes referred to as a strap region. So if I use that terminology, that's what I'm referring to. And one of the things that has not been looked at that's very relevant to this whole idea of the multiscale mechanical interactions and the simulations we're doing is the boundaries of these gels have not been considered in how they're influencing the development of this straps or fiber alignment in these explants, or how these spatial patterns of explants are affecting things. And so what we're looking at right now is what happens when we alter those boundary conditions. So we have these fibrin gels. We're sticking with this triangular explant pattern for the time being, and we're casting these gels with the explants in these square PDMS molds, and then what we do is we keep them attached to the molds and call that our fixed in-plane boundary condition experiment. Or we basically remove the attachments of the gel with the borders of the PDMS mold. It's still attached to the glass, and that's called our free in-plane boundary condition. And then what we do is we also make these gels with beads, 4-micron beads, to help us track the gel displacements. And we take tiled images over a 48-hour period so that we can look at the short-term remodeling that's occurring here, which really is only involving structural reorganization. It's too early for any appreciable compositional changes to occur. So here you can see a collection of tiled images of the 3 explants contracting the gel with the fixed in-plane boundary conditions. And there are all these little dots in here that correspond to the microbeads. And so the beads, it actually turns out, make it difficult to actually see the fiber alignment develop. Probably they're diffracting the light. But you see matrix densification that's occurring in the same triangular pattern. And what we can do is look in this densification region, pull out one of the single frames that we have, and this is just showing the explant axis. So this is where the strap region is. And we can watch how those individual beads are displacing through the gel as a function of time. And then we have some image processing algorithms that we use that allow us to determine the displacement fields in the X and the Y direction, which we can then convert into something like maximum principal strain. And so this is showing what's happening in the fixed in-plane boundary condition experiment. This is the same experiment with the free in-plane boundaries. And what you can see here is that there's much more consolidation of matrix occurring, and you can also see that there are cells moving along these regions of alignment. There's also this weird pulsing thing happening here that I think is very interesting, but I'm not sure if it's repeatable or not yet, so I won't speculate any further on that. As we all know, when we do these kind of experiments, sometimes something odd like that will happen from time to time. But the overall pattern of reorganization is consistent. And so, again, you can pull out the individual frames and quantify the displacements and get measures of strain. And we find that we get much larger displacements and strain in the in-plane boundary condition situation. And we can make comparisons here. These are just histograms from the 2 conditions, and we see that we get, on average, much larger strains with the free in-plane boundary condition. We're still analyzing this data and putting it together. We want to try to also correlate this to what the loading would be. It's a little bit harder problem, so right now we're focused primarily on the strains. Another component of this that I just want to touch on is that we also want to measure what the forces are that the cells are collectively generating. And so what we can do is take our gels and put them between our grips in our mechanical testing system. And then if we use an extra long working distance objective, we can watch at least what the cells in the top layer are doing as we collect the load that they generate from isometric tension. So we're not displacing the sample, we're just holding it fixed in place and letting the cells compact the gel and measuring that force. And one thing I just want to point out is the shape of this curve. I'm going to comment on that a little bit later on in the simulations. Okay, so what we've got are these experiments that we're doing right now with these explants as a simpler system to get started. We're doing this time-lapse imaging. You can see the fiber alignment I was talking about before in this image very nicely. And what we want to do is develop these multi-scale simulations so that they can tell us more about what's happening in these experiments, as well as develop the physics of the problem so that we can use it eventually as a predictive model for wound healing. So what we've got here is a 1-element thick finite element mesh that's got 576 trilinear hexahedral elements. And what we are doing here is partitioning the model into 2 different types of domains. We've got cellular domains, which you can see as the blue regions here, and then the pink represents the fiber network extracellular matrix domains. And we're treating both of these the same way as the same 3-dimensional fiber networks that I showed you in the simulations from previous work. The difference is that what we're doing is simulating the effect of the cells compacting the gel on the surrounding matrix by taking the cellular domain networks and incrementally shrinking the reference length of the fibers a certain amount so that it effectively creates tension in the surrounding fiber networks. And this is really just it's not meant to represent what's actually happening with a cell. It's a starting point for just trying to get a sense of how those cells are affecting the remodeling in the matrix around them. And here you can see just what those fibers in the cellular network are doing under those rules. The fibers were shrinking the reference length. They're connected to each other, so there's force equilibrium, they end up generating tension in the surrounding fiber networks. So these are simulations for similar parameters to what we've done experimentally, where we're looking at free surface boundary conditions and fixed boundary conditions. Those are where the cells are located and then the surrounding matrix. And here you can see what the strain fields look like for the two different simulations. And you can see that we get the triangular pattern in the strain field that is reminiscent of the triangular pattern of the fibroalignment that we see experimentally, and that there's a difference between the two. This is just showing screenshots of what I just showed you for the fixed boundary condition case and the free boundary condition case at 20% change in the reference length of the cellular networks versus 40%. There's some white arrows on here that are showing the first principle strain as well. This middle region is the uniaxial condition, where the lateral sides are free, but the top and bottom are fixed. And we simulated this too because one of the things we're interested in is if you deform a gel like this, how is the load transmitted differently in a situation where you're accounting for the cells compacting the gel versus when you are not? And this is relevant to tissue engineering practices where if you're trying to improve the mechanical properties of a tissue like a ligament or a heart valve, it's common to mechanically condition them because it will trigger the cells to produce more collagen and make that construct stronger. Okay, so what I'm showing you here now is just some network level simulations for the locations indicated in the circle. So here you can see for the fixed boundary condition in the center region here, which doesn't have a lot of strain, there is some fiber force that develops in these networks, but there isn't a lot of reorganization when you compare it to the type of reorganization forces that develop for a network that's now along this axis of alignment or this strap region. Okay, so another thing you can look at is the change in fibroalignment, which is more synonymous with what we're measuring. Well, we're also measuring strains, but this is sort of more synonymous with the visual cues of fibroalignment. And what this is a measure of the strength of fibroalignment and how it changes with reference to what it was initially. So it's the change in fibroalignment. And so you can see with the amount of compaction that's going on, there's a big change in fibroalignment that again is forming this triangular pattern and also has these offshoots that are directed towards the boundaries, and we see evidence of that experimentally as well too. We can also play around with the number of explants, their spatial relationships, how far apart they are, and see what we're getting there. And we, by the way, I should say we are doing we're in the process of doing experiments with similar, 4 explants, 5 explants, and things of that nature. Going back to this other idea that's a little bit of an offshoot, this stretching these gels to see how it alters the distribution of loading in the mechanical environment when you take the cells into consideration, which is something that many people don't do in these simulations. What you see on the left is the force stretch ratio diagram, where the stretch ratio refers to the whole tissue. And this inset is showing basically the amount of force that's generated for the case where we have 3 explants without any stretch. So this is how much force you find on the top boundary just during the compaction phase before we stretch it. And the thing to notice here is that we've got this exponential increase that's occurring here that is the opposite in shape from what we see experimentally, where we have this rapid increase and then plateau. So that's a good indication that there's quite a bit of work to do on representing those cells and how they're interacting with the matrix. This larger part of the plot here is showing you the overall force stretch ratio response as we stretch these things. And you can see that with the explants, there's a small difference, a higher level of force in there. And then what I find interesting is that the patterns of the mechanical environment are, of course, different between the two. You have some regions of high strain here and some regions of low strain and compression there. And if we look at the 2 models, one of maybe I didn't make this clear enough. This one doesn't have any explants. So this is if you just try to model the tissue without accounting for cells. And what that means is we've got we've got the same fiber networks everywhere. We're just not treating the ones that are explants as actively contracting. They're just now reverting back to being extracellular matrix networks. If we look at the difference between the no explant and the explant and see where we're seeing large differences in the strain field, the regions with dots are giving you an idea of where that is, and it's not a surprise that it's around where the explants actually are. And then this is just showing the fiber strain distributions for the regions where we have the dots, and you can just see that the profiles are different from each other. So we just received this proposal, the award for this proposal in July. We're making a lot of progress, and you're seeing what we've done so far. I just want to talk about some other stuff that's ongoing and some future work. So one of the things we want to do is improve the precision of the placement and volume of the cells of the explants. So we've developed a micro-manipulating device that allows us to do that. I had an undergrad who did I think a fantastic job in bringing that into reality, and hopefully you can see how nicely those are in comparison to what I was showing you initially, where we were hand pipetting things. We are also in the process of exploring other explant geometries and boundary conditions. Another component of this project is to move into not only doing this with the DIC that I've shown you, but to supplement that with information from multiphoton imaging so that we can look at the three-dimensional structure and the changes that occur there as well. And this is actually just a reflection mode image of one slice from a stack where you can see the cells here and the fibrous nature of the gel there. It'll allow us to quantify the reorganization that's occurring through the thickness, as well as help us construct hopefully more accurate fiber networks than the idealized ones that we're using now. I haven't touched on it yet, but the sort of other impetus for this proposal is to quantify the remodeling process that's occurring. And so, if you look at this schematic, over the 48-hour period, we have these blue lines that are supposed to indicate fibrin just becoming aligned and forming this triangular pattern. What we expect to see from previous experience is that in about a week, we should see the cells have migrated along these regions of alignment, and that there's collagen deposition that's occurring that will increase and mature with additional time. And we're in the process of getting our fluorescent probes worked out to look at that. And then we want to do things like deform that environment and see how it alters the remodeling process. And the schematic here is showing us doing this at 2 weeks, but another major point of the proposal is to do this at earlier time points to see, is this pattern that develops sort of locked into place beyond a certain point? Meaning, if you let a wound go for a few days, can you alter that mechanical environment and still get it to remodel a different way you want it to, or do you have to get to the wound site sooner than that? And then as far as the remodeling or sort of the computational modeling goes, one of the things we're working on currently is to still use this sort of crude approximation of the cells compacting the gel, but change the rules from being displacement-based to force-based to see how that alters things, because I think we can all agree that, or hopefully all agree that a force-based measurement is probably more realistic of how a cell responds to its environment. And as we're doing that, we want to start to modify the network structure, incorporate additional physics in the model, and then focus on trying to develop better approximations of modeling the cells and their interactions with this matrix. And so sort of the overall benefits of the proposal then are that when we get this all done, our goal is to have this new model system for developing a more detailed view of the wound healing process, and it will improve our ability to devise and assess new treatments for managing scar formation. I've only really talked about the mechanical component of this, but I think you can see that other avenues, once the system is sort of developed and established, would be to look at how different drugs and biochemicals can affect the remodeling process, and how maybe combining a whole bunch of different stimulatory cues could be used to manage the wound healing process. And so with that, I want to acknowledge my sources of funding, especially the R03 that I'm talking about today. And I'd also like to acknowledge people that have been involved in various aspects of this project. Down below, you can see my graduate students, particularly Arabit De Jesus, who's done all the microscopy and experimental components of this, and Maziar Agvami, who's been involved in all of the computational simulations. And I also want to acknowledge Victor Barocas and the University of Minnesota, where I was a postdoc for several years, and where the previous work was done that I talked about in the beginning of the talk. So with that, I think I'll stop and see what kind of questions people have.
Hi, Ed. Thank you for a very nice presentation. It was a beautiful talk. So we'll open it up for questions at this point.
So,
sorry.
Is your model very sensitive to mesh quality, the finite volume size?
It is. There is some sensitivity. So we're looking at that right now. What I can tell you is with the previous work where we were just looking at the collagen gel system when I was at Minnesota, we did refine the mesh to a point where we weren't getting substantial changes in the remodeling response. And so the mesh sizes I was showing you were adequate for that particular problem. I think that what I expect we're going to see as we look at this in a little more detail is with the new cell compaction part that's making this nonlinear problem even worse, that we're going to need to refine the mesh, particularly in the regions around where that's happening.
Mm-hmm.
So.
Okay. Thank you.
I have a question. This is Del Jaron. I don't know, my picture disappeared. Can you hear me?
I can hear and see you.
Oh, okay. I cannot see myself. Actually, my question relates to a statement that you made at the end of the presentation. I was going to ask you that question before you made that statement, and that is, you've been looking only at the mechanical properties.
Right.
That's the basis of your model. But clearly, when you're going to deal with wound healing, you will have to also consider some of the biochemical processes. Do you have any ideas how you would integrate those processes into your multiscale model?
Yeah, so when you have the platelets degranulate, and I'm just saying this since we're recording this for other people who are maybe not aware, they dump out a bunch of cytokines and growth factors, TGF-beta, PDGF, things like that. For the sake of establishing the model right now, we're not adding any defined growth factors. We're just using fetal bovine serum. But once we've got things sorted out with the system at hand, one of the things we want to start adding is TGF-beta into the system, because when I was at Minnesota, and I didn't talk about it, but with the fibrin remodeling from the tissue engineering standpoint, TGF-beta is added in order to enhance the amount of collagen that's getting produced. So I have plans to incorporate various aspects of those types of biochemicals into the system to see how they modulate things. Of course, that's still going to be relatively simplistic compared to, the complex profile of these different chemicals and their time dependence and their interactions with many different cell types. But it's definitely important.
How would you link those processes to your model, to the mechanical model? I mean, the linkage is going to be very important.
So are you talking about things like actually incorporating diffusion and discrete particles that you would maybe call growth factors, or?
Could be, that's correct.
So that's certainly something that can be incorporated into this framework. I haven't tried to bite down on that particular thing because I'm just trying to focus on the mechanical part. But the framework should be accommodating towards incorporating those kinds of physics into the model.
Thank you.
I mean, you could also experimentally, and I'm just kind of talking off the top of my head, but you can set up gradients in these gels where you introduce growth factors on one side versus another, and look at things like that as well. So, I mean, I think, with some more thought, trying to do experiments that kind of delve into those kind of details are certainly doable.
That would be a new challenge, I think.
Oh, it certainly will be a challenge, but yeah, one thing at a time right now.
Thank you for a nice presentation.
Very nice.
Thank you. Thanks.
So Ed, I probably missed it, but I noticed that you mainly presented the spatial predictions of your model, and at the end you mentioned that you would want to look at time points at 2 weeks and things like that. So I was just wondering how well your model will predict the temporal changes with respect to the data. And what is the time course for the remodeling?
So right now, the model as it exists is not actually incorporating any kind of compositional changes. It's purely structural. So linking that to the experiments at this point, we're only focusing on the 48-hour period because that's all, for the most part, structural changes. In the past, we have implemented rules that maybe would describe how fibers could degrade, which would be something that we could start to incorporate too. But the big challenge is going to be coming up with reasonable experimentally based rules on how collagen production initiates. And so, you know, there's some ideas about that that are being developed as well. But right now, the experimental side of things is kind of more out in front on the remodeling side of things than the computational side.
Okay, thanks. Any other questions? If not, I want to thank you, Ed, for your very nice presentation on behalf of IMAG and the consortium. Look forward to your progress and your contributions in the consortium.
All right, thanks very much.